So I'm going to try and stick to the time. Thank you for being here so early in the morning, at least for the people who partied last night. And thank you for Betty and the planning committee, which I'm part of, for allowing me to give you a talk about this particular topic. I will try and split it two ways. So I will give a very brief overview about using phages as surgical applications, which basically means anything that's going to go through the skin. And then I will also talk about what we've done in our lab with our group for the last 3 years. Okay, so as you may or may not know, animal models is basically also known as translational research, and it bridges the information from basic science through to the final application of treatments in clinical practice. Animal models have been very useful in contributing towards the mechanism of finding out how diseases work, as well as finding and helping predict how drugs and treatments may also work in humans, although not always the case. It is really important, however, to make sure that you perform a systematic review and meta-analysis of the published work so that it can help improve the experimental design. So, phages as antimicrobial agents. Why? Well, as we all know, there's been an increase in antibiotic-resistant bacteria, and to name a few, we've got Klebsiella pneumonia, which is normally a commensal in the gut, but has evolved to be part of the carbapenem-resistant Enterobacteriaceae. It is very problematic in patients who require indwelling devices. Another organism is Enterococcus faecium, and that is also another commensal of the gut. Very problematic in that it can cause bacteremia, meningitis, got a few urinary tract infection, which later on Susan might touch upon. And then my favorite, because I work with it, Staph aureus. Actually, the very first Staph aureus resistant to penicillin just came about 3 or 4 years after the mass production of penicillin. And as you can see, there was a very sharp rise in resistance to penicillin drugs, related family of the drugs, from the 1980s onwards. And very interesting for me, I found, is vancomycin resistant Staph aureus is actually being found to occur as a conjugational transfer of the Van-A operon from a VRE strain to Staph aureus, making it resistant. So just a few beneficial characteristics that we'll look at or using bacteriophages as antimicrobial agents. Hopefully you can see the blue ones because those are the ones that I kind of like the most, especially since the microbiome has had a whole amount of work being done, and lots of publications came out last year and this year about how we can try and not disturb our good bacteria. So that's kind of how I always try and sell it to the students who are interested in doing research with me. It's like, well, you know that there are good bacteria and bad bacteria. Same thing with viruses. There are good viruses, like bacteriophage, that are lytic, and bad viruses. Okay, so moving on, just a few examples, 3 of them, on specific models that have been used of bacteriophages and their application, in essence, surgical applications. So first of all is treating carbapenem-resistant Enterobacteriaceae, which is Klebsiella pneumonia, as I mentioned earlier. And what they did is Kumari et al. In 2009 paper, sorry, it's not very clear. So what they tried to do is they made a burn infection model and injected the bacteria. So it's a lethal dose of 100, meaning that all the animals are going to die after 72 hours, actually, if you inject this particular concentration of bacteria just underneath subcutaneously underneath the wound burn that they made. And what is really interesting about this particular study for me is that they tried to use a cocktail of 6 phages, I think it was, and each individual phage and its treatment towards the infection is shown here, and this is the bacterial load found in the skin that's been later explanted at different time points. So there's 24 hours is the most darkest one, 48 is the white one, and then the well, it's not multicolored, but striky one, I guess, is at 72 hours. But what's happening is the bacterial load at 72 hours is the lowest one with a cocktail, which I thought was really interesting, although it's pretty good with this one as well. So in this pinkish block highlights the control, so that's the infected, and what they do is they compare the treated versus the untreated. And this is just, hopefully you can see a little bit, not great resolution, but this is how the wound burn looks like and how it is after being treated at 72 hours. And then this is the next guy is just technical problems. Just how a mice normally looks like when he's nice and healthy. So following on from that, this should be Biswas et al. In 2002. And what they tried to do is they tried to treat vancomycin resistant enterococci in a bacteremia model. And the most interesting bit that I found was they found that it to be dose dependent. So there was just one phage, and the really good, in my opinion, study design here was that they actually used long circulating phages to do this study. Whereas a lot of us tend to just, get our phage, isolate it, purify it, and then try it out that way, which you'll find there can be some problems afterwards. But anyway, so it's dose dependent, so that should be 3 times 10 to the 9, 8, and it goes down. And what they found is that if the bacteria is added, the phage is added immediately after the bacteria, then, or 45 minutes after, I should say, then at low MOIs, MOIs of 0.3 and 3, you get really good results. That means 5 out of 5 of the mice that they treated had recovered, survived. And then they also did some they found time-dependent rescue using the high dose, right? So there's 5 hours post the infection and then treated. There's 18 hours, and then there's 24 hours. Now with the 24 hours, they found that they did lose some of the mice, but after 5 hours, they actually managed to get, again, 100% survival. 100% meaning 5 out of 5, right? So I thought that was really interesting. My computer loves going to sleep on me. So I thought that was very interesting. Okay, so lastly, another example is Caparelli et al., which is one of my favorite papers. And again, they used long circulating phages. They first found the phage that they wanted to use, and then passaged it through a mouse that they actually infected, but in very low volumes of bacterial load, so that they could manage to obtain phages much later on after the initial phage load. So what they found is that if they used an MOI of 10, all of their mice survived at day 20. It should be day 20. And they also found that when they were trying to treat abscesses, so this is more of a local infection, what they've done is they subcutaneously inject the bacteria in one flank, and then the phage followed immediately after that on the other flank, and they stopped the abscesses from growing, right? They inhibited the abscesses by immediately giving that phage administration. However, when they delayed the abscess, meaning so they added the bacteria, allowed the abscess to develop for 4 days, and then treated it. They didn't stop the number of abscesses so much, but they did reduce the weight and the bacterial load of the abscesses. Okay, so what have we been working on? Well, well, you may not know, but I'm from the Department of Orthopedics, or I'm based there, so we're always interested in bone, and I'm a bacteria person, so I love infections. So put those together and you get bone infections, and that's what I worked on, basically. So the idea was really for me, I was really interested in trying to see the proof of concept whether we could use phage therapy to treat or prevent bone infections. First of all, what I thought is, well, do I really want to go through the isolation and all that? Because that's going to take time. I mean, and of course, we want to get our own phages and isolate our own phages, characterize them and purify. But because we really want to start the work as soon as possible, I thought, well, I want to use a phage that is well known already in the field. So I decided to use Staph Phage K. I then tested Staph Phage K, or I didn't actually do it. I love my undergraduate students, so I put them all to be famous there. This is Zach who did this particular work. So he actually titered the Staph Phage K on its propagation host, which is 19685. So if you buy it from ATCC, you should get that strain. And then tested it out against around 30 clinical strains, which all of these are MRSA, so most of them were MRSA. There's quite a few that are Staph aureus, and then I had some reference strains as well to make sure that other people could try and reproduce the work. And then what we also wanted to do is actually use bioluminescent bacteria, Staph aureus in this particular case, so we could use our fancy new equipment. And as well have a look at the infection in real time and what's going on without trying to kill so many of our little rats. So as you can see, Staph Phage K was pretty good at a lot of MRSAs. It looks roughly 50, and this is just a random number of clinical isolates that we obtained from a wound care center. Okay. So Kelly, who's here, say hello to the say hello to her when you see her around. And Lizzie also worked on this particular piece of the study, which it was actually being able to quantify the bacteria present by both looking at the viable cell counts and the metabolic activity using the IVIS machine. So I wanted to make sure that we got a really good correlation between that. So I'm a numbers person, so I kind of like it to be. And as you can see, it's pretty straightforward. So with another of the animal studies that I'll show you later on, what we did is we used a roughly 1 × 10⁷ CFU, which is along this end. So this is the bacterial load, and what we end up is we want to make sure that we're going to get roughly between 4.5 and 5 photon counts. So that's what we're looking for, just to make sure that we're getting that amount of bacteria present. Okay, so the pharmacokinetic study, first of all, we wanted to see what our dose regime was going to be, or how much concentration of phage we were going to add. So what we did initially is to try different routes of administration, as you can see, intraperitoneal, intravenously, subcutaneously, intramuscularly, of our purified phage. We actually sent our phage to be purified by Frank and Jofal, I can't say his company very well, sorry, who really purified it and had a look at the endotoxins present and so forth to make sure that it was a really good product. Because what I've actually given a talk to immunologists, so the first thing I tend to get is, well, are you going to get a lot of immunogenic occurrences and so forth? So wanted to make sure that our phage preparation was really pure, or as pure as possible. So as you can see, so the intraperitoneal injection did give us, so they all had exactly the same, from the same batch of phage preparation. So they all had 200 microliters, one-time deal, and then we followed them for, actually it was 48 hours, but we found that after 12 hours there wasn't much phage around. It was very hard to detect. And we used exactly the same rat, and we took out samples. This is why we ended up using rats, is in order to collect the same blood samples from the same animals. Although the IP gave us the highest phage counts, the intravenous administration was the most consistent. So that's kind of what we went with. And then this is just to show what happened at 48 hours when we finished when we killed the mice, and most of the phage we found in the spleen. So the other thing, come on, wake up. So then the next two graphs, what I'm showing is, this is what happens with an infected, or a few infected rats actually. So this is 9 infected rats, and throughout time, this black line over here with the standard error means are basically this graph superimposed onto this other graph. And as you can see, the phages are being detected long after because of the bacteria being present there. Next is we tried multiple dosing. And you can see the arrows then are showing, so we added the phage pre and post surgical application, and then the bacteria was added after the surgery. And then the phages were added 3 days after that. And the really interesting information that Peter and I found was that the phages, although they were still being isolated, obviously I'm adding new phages, so I'm going to see them, but they're at lower numbers. So they seem to be maybe being removed from the body. Okay, so Peter did all of the surgical procedures in our rats, and you might not be able to see these so good. Hopefully you can. There's a little hole there, which is what we do in the tibia, and then we add the bacteria, and then we make sure that the bacteria is nice and active. And our endpoint is 14 days, where we are seeing osteomyelitis occurring, so deformity on the bone. And then these graphs just show, as every day the rats get monitored for the bioluminescence. And another interesting point was that with the phage treated, running out of time. With the phage treated, there seems to be a bit more metabolic activity going on than the one that's not treated. And these are just, again, students and research staff that are helping us with the study. So, second to last slide. So it didn't work, basically. Going back to the drawing board, where I was thinking is, oh God, how can I actually find out how we can work this instead without going through so many animals, because poor little blighters have to get killed at the end of every time we study them. So I tried this combination therapy. But what we ended up doing is going back a little bit to our models, and we actually used biofilms grown with the CDC bioreactor, so the nice dynamic and hardy biofilms. And before that, we tried phage cocktail done by Kurt, made up by Kurt, and he actually tested out phages that or the students, I should keep saying, it's the royal we, of course, used isolated from piophage and intestiphage product, as well as some sewage water. And what they found was that there was 5 of the phages from those Georgian products that were pretty good and a bit better at some of those clinical strains in comparison to phage K, which is actually highlighted in the black box. This is actually shown in the poster, so you can have a look at that and ask Sija plenty of questions, because this is mainly her work. So when we compared the untreated biofilm against the treated ones, there was a significant difference, but there wasn't any significant differences between, time's up, the different concentrations of phage, K on its own, and the cocktail of phages. So not so great results there. So going back to the drawing board, I thought, what about if we just mix it with antibiotics, right? What happens then? So just to show you where to look there, this is 10 seconds quickly. Untreated at the top, vancomycin and phage second row, phage only third row, and vancomycin only last row. And you're probably thinking, well, there's not much difference between the vancomycin and phage and the vancomycin only. And looking at the bioluminescence, in essence, or the metabolic activity, yes, that's true. But if we actually look at the bacterial counts, what we found is that there is an increase of planktonic cells, so that's the cells dispersing from the biofilm onto the media. There's an increase at the 72 hours as comparison to the biofilm. I'm running out of time here, I'm being told. But however, comparing the vancomycin only and the vancomycin and phage, there is a much greater significant difference in the bacterial load on that biofilm. And then lastly, just my funding sources. Phew. Right, and while there's questions, I'm plugging an article. It's open source, so please feel free to download and everything, because normally you have to pay for that journal. Thank you.